And we're going to introduce our seats. Good morning, everyone. Thank you so much for joining the last plenary of this conference. We had some amazing talks, amazing plenaries, and today is no exception. We'll continue on with adaptation symposium today, linked to this plenary. So if you find this topic interesting, stay in this room. I have the pleasure today to introduce Kat Valero. I will tell you a bit more about her, and then I will later tell you about her amazing talk today. So Kat did her PhD with Miguel Bences on Madagascar frogs. After that, she did a postdoc in Harvard with Jonathan Lysos on anolis lizards. She had 3 faculty jobs, and she's currently at University College in Dublin, in Ireland. She enjoys it very much. And recently, I would like to congratulate her because she has been awarded the ERC Consolidator Award. So very congratulations. So I look forward to this talk. I'll hand over to you.
Thanks, Natasha. Okay, I hope you can all understand me well because I talk enormously. Trying to use as many microphones as I can. First of all, thanks everybody for inviting me, especially Natasha, Simon, Mark, and the other members of the committee. I'm super happy to be here. It's been quite a while since I've been on a HERP meeting. And also, you've heard that life's kind of knocked me around a little bit to different places. So I met different people, and I also worked on lots of different organisms, not necessarily reptiles and amphibians. And in the process, I kind of learned a lot about genomics and related to responding to stressors of the abiotic environment and also to adaptation. And I've tried to kind of, over the years, to merge all of this knowledge together. So I will talk about many different organisms, and if you kind of get confused or if you need have some additional help following what I'm talking about, there's always going to be a little picture of the species that I'm talking about in the top right corner of the slides. Okay, the second thing that I just wanted to mention is that adaptation can mean different things to different people. So humans typically, when they talk about climate adaptation, they mean the built environment and any sort of measures they can take to kind of prevent exposing themselves too much to the climate. And when you talk about organisms, it can mean also changing behavior on an individual level, but also it can mean adapting on an evolutionary level. And I'm going to talk mostly about the latter part. So I'm going to talk about functional and comparative genomics today. Genetics lecture at 10. Okay, so first of all, I'm going to outline a little bit what the responses of organisms are when the environment is changing. So what can they do? Then I'm going to talk a little bit about my and colleagues' research on the genomic basis of adapting to climate. So what genes are being used, what functions do they have, are they different or the same in different organisms? And then it's going to be a little bit more theoretical and sort of experimental. I'm going to talk about the importance of functional genomic networks. It might be new to some of you. I'll go through it carefully and hopefully we'll learn something new after that. And then lastly, I'm going to just very briefly conclude by outlining what we should study in order to keep pace with the environment that's changing and our knowledge of it in the process. Okay, so let's have a quick look at responses to changing climate. Okay, there we go. So I really like this graph because it combines several lines of information. So it has the IPCC predicted pathways of how our climate is going to change in the next couple of years. And it also has all of the values of the average temperature during the time after the Industrial Revolution, but it also shows everything up to 65 million years ago when the primates evolved. And something that I find really interesting is, I've tried it here and there in different talks, but I just wanted to show it here as well. If you're following those different pathways that are predicted backwards, the RCP 2.6 pathway actually brings us back about to the climate that was last experienced by Earth about 15 million years ago. But if you then if you're thinking that we're not going to manage the carbon emissions and it's going to be the 8.5 pathway, that would actually bring Earth back to a climate that last has been experienced by organisms about 45 or 50 million years ago. And you can see that was the time when apes and monkeys evolved. Or, if you want to think more herpetologically, that's about the time when the two major groups of lacerted lizards evolved. So the Araneadini occurred about 55 million years ago, and then speciated through this hothouse period, and many species kind of arose here. And then later, the Lacertini, they evolved as the climate was kind of more pronounced stable period. And then subsequently, however, in the Kenozoic, overall, there's been a trend of cooling. So I'm always asking myself the questions, if you're looking at adaptations to heat, yeah, you can look at different organisms and live in hot environments, but maybe, many of the organisms that we have around today have been just experiencing cooling climate for such a long period of time. How much potential is there for adapting to things going the other way around? Secondly, I think that's not news to anybody. We're not just looking at the average global temperature, but we also have to look at extreme events, which are becoming more prominent as we speak. And something that I always find interesting is that we don't really have any good data on these kind of mass mortality events that occur during heat waves. I mean, right now we're in a heat wave as we sit here. It's been about 5 days of really above average temperature. And some of the estimates that are coming in are actually not really scientific publications, but people that just estimate, like going out in the field and looking at the animals and estimate like how many have perished. And there was in 2021 a heat dome in the Pacific Northwest on the coast, and there was an estimate that on this rocky seashore about 1 million seashore animals, including those blue mussels, were just cooked alive with 53 degrees Celsius temperature there. So what responses can animals show if things like that happen? And I've just kind of, I draw this mini comic like a while ago, so it's not that great, but if you want to see the same thing a bit better, you can look at this recently published review, not by myself, other authors, about different responses that can happen. So on an individual level, if you can imagine there's maybe a hot day, and we have this grumpy looking rain frog here as an example organism, maybe not the best one, But anyway, first thing that happens is that it will activate its primary stress response, so it will activate chaperone proteins to protect its own proteins. It will activate the stress response. It will, in the process, consume ATP, so the energy reserves are going to be depleted, which means that it maybe then has a bit less energy available to invest in other things that it needs to do. Then if the stress persists, so perhaps we have a heat wave, the next thing that can happen is that our organism will change its behavior to thermoregulate. So in this case, I just put the rain frog in a little burrow. But some organisms that are kind of adapted to experiencing unpleasant conditions once in a while, they have the ability to lower their metabolic rate. So, hibernation is an example for cold temperatures, and estivation would be the equivalent for hot temperatures. So we'll just lower their metabolic rate. But the problem is every time they do that, they get in and they come back out. They will produce oxygen, reactive oxygen species that will be having to take care of by the stress response as well. The other thing that organisms can do is they can change their behavior to migrate out of the area. And that is on an individual level, literally just dispersal. So they will use their dispersal capabilities to get out. The stress response phase 2 is like this change of metabolic rate. And then if we think about the next generation, they can apply plasticity, so they can either they can change their epigenome, and that will then kind of more permanently change the way the stress response operates. You can have some clutch members here that will perish either in the embryonal period or later in adult life, and some of the ones with epigenetic modifications that make it another generation or 2 or 3. We don't really know how long epigenetic plasticity lasts once the stressor is removed. That can be a good thing or a bad thing. So typically it's been thought of as a good thing, so it buys the population time to kind of sit out the bad period. But it can also be a bad thing because it takes away the capability of them to experience proper adaptation, so selection on existing variation. And once the epigenetic signal goes away and the stressor is still there, then that's kind of the end of that. But it could be that there's some beneficial variation, like the yellow eggs over here, that then will experience an increase in allele frequency in the population, so that you have then in the end true evolutionary adaptations. We have a change in traits. And this review helpfully points out that this change in traits can be either traits that favor persistence in this new environment, such as changes in physiology, or it could also be traits that favor getting away from the environment, such as migration traits. All right, and because I'm a geneticist, I'm obviously going to be on the whole change in gene sequence part of it. Okay, so in 2008 already there was a seminal paper that said the same thing, which is that the rate of adaptation will primarily be set by the rate of microevolution. Microevolution is defined as a change of allele frequencies, and that's why I've, a long time ago, got really interested into looking at what kind of genes are organisms using or modifying to be able to kind of change as the environment changes. Plasticity is really interesting, physiology is really interesting, but my personal area of interest has always been genetics. So I set out to look at some of this in several studies. So I'm going to start really early, so that's like blue-eyed young me, thinking that the easiest way how to find out what is the genomic basis of climate adaptation is go to the field, sample cold and warm adapted populations, and detect genomic outliers. So that was the thing back then. We're looking at GWAS, we're looking at SNPs and things like that. But then immediately I realized that the problem is that you find a whole lot of SNPs and genes, but not all of them might actually be causally related to the change in the environmental parameter that you're studying. So it could be like changes in sexual selection, changes in advertising traits, changes in predation or prey, and things like that. So there could also be just population genetic processes, such as genetic drift. So you will have a lot of kind of dots appearing that aren't really related to your stimulus. And then when I was talking to people like around that time, so it was probably 2014 or 15, they were all saying, but we already know how this works. Well, we have feature protein 70. That's the universal chaperone, and it always comes on when there's heat stress. So actually, we don't really need to know much more because that's what we study. It always comes on in its way. and it is great. But the problem is with feature protein 70 is that the sequence is highly invariable. So you can look at a HSP70 from a yeast to a human, and it will be very, very similar in shape and form and function. However, there's recently been some studies that show that the expression levels of feature protein 70 are actually varying depending on how well adapted the organism is to heat stress. So there might be other genes that are being modified that then modify the expression level of this very general response protein. So what we thought we were going to do is just kind of take a bit of stock approach. So what genes have been published in the literature to be known to adapt to differences in climate. We use vertebrates and focusing on ectotherms. There wasn't that much about lizards or reptiles, but there was lots of kind of other species around. And then we also kind of took stock of what physiological adaptations do animals experience when they adapt to different environments. And then what we did is we just took the genes, we looked at the functions of those genes, gene ontologies. I'm going to mention this quite a bit. And then we matched that up against the functions that we know to occur from physiological adaptation. And basically we said, okay, which genes are actually related to those functions that we expect to pop up in the phenotype. And that's going to maybe help us identify in the whole entire genome with all of the outliers and everything, the ones that are functionally related to difference in temperature. So it was 175 papers, and we came up with this. So this was a bunch of genes. That is a genetic network that shows how genes interact with each other. In the middle, you have the gene symbols, and then the connections are like what functional interactions they have with other genes. We came up with a bunch of genes that were actually quite prominent, and we looked at the functions that they have, and we came up with this set of 8 candidate functions for thermal adaptation. Okay, so it was a very basic kind of just overview thing. And if you look at those functions, such as lipoprotein metabolism is quite special, but things like membrane transport is really general. So you could immediately go and say, well, probably all of the genes have any of those functions, but that's actually not the case, and we're going to go back to that later. And the second thing that we found already in that study is that there was quite a high degree of similar genes in totally different species. So maybe like a Drosophila sitting in a hot spring uses the same gene to do that adaptation compared to its sister species not in a hot spring that a fish would use for adapting to sulfur springs. So that was already kind of a bit of what's happening here. Anyway, that's supposed to be over here. Set out to test the hypothesis by collecting Anolis cybotis from the Dominican Republic. Anolis cybotis is also called the trash anole because you find it literally on every single trash can on this entire island. And you can also find it in all the natural habitats. It doesn't matter if you're at sea level in 40 degrees heat, or if you're like on the very, very high lands up there, about 3,000 meters high, and sometimes it freezes in winter. You can see that picture here of the ice forming on that vegetation here. So it goes from very hot to very cold, and we just looked at, well, what kind of genetic differences do we find, the phenotypic differences. So the nice thing about the Dominican Republic is that it has several mountain chains and then lowlands intervening, and they're all colonized by the species, but different populations of it. So we looked at, is there any phenotypic differences every time you go up versus if you're down, and we found some on X-rays. So we found every time you go high up, you find lizards that are quite stout, flat, wide bodies, short limbs, and a lot of bone density. And if you go down, you find lizards that are slender, long-limbed, and have less dense bones overall. And then we did RAS sequencing, which probably most of you are familiar with, just to look at what kind of genomic data supports this. But first of all, we saw a population structure just to use as a covariate, just to make sure we're not getting confounded by that. That's not really that important. And yes, we found a lot of SNPs that were different. Interesting pattern that we observed is as you go up with elevation, you have a parallel change of osteomorphology, body condition, and also the proportion of rare SNP alleles. So the rare alleles kind of increased towards the higher elevations, and because you are a tropical lizard, high elevations aren't really that common in the Caribbean, and neither is the climate associated with them. We were hypothesizing that we're looking at adaptation to cold here, not to heat. Most of the SNPs we found actually weren't in protein coding genes, and I always wanted to analyze the data set, but I never had the time to do that. So they're kind of being put on the slide. They were in microRNAs and transposons, which is interesting in itself. Remain 14 different proteins. It says 14 SNPs, but that's wrong. There were sometimes 3 or 4 SNPs in the same protein coding gene. And they are also shown again here with the functional genetic network. And in green, we have the ones from the study where we tested. And in black, we have the genes that we predicted to be important for climate adaptation beforehand. So yeah, those are not the same genes. However, they are functionally really closely connected. And you can run statistics on the entire genome to say, well, is this functional association closer than you would expect by just drawing random genes? And yes, it was. So 17 of those previously predicted 48 genes for thermal adaptation were also in this functional network in black. Okay, and then we took the list of candidate functions and we were just kind of looking at, okay, which of those genes actually have the functions that we're looking for? And 7 of the 8 ones were covered by those genes. So that was good because it told us that yes, this difference that we observe across the different elevations possibly has something to do with adaptation to climate. A little bit more in detail, we also were interested in finding out like where these genes are expressed. And now this is where the confusion comes in because it's data from human expression. So I'm just happily jumping between organisms all of the time. Might not always be the best or the correct thing to do, but it's the best thing we can do with the information that we have. So I think it's totally fine to do it. So we have expression from the human where those genes are expressed, and it comes out the endocrine system and in the brain and during development. And interesting, there was a study a year afterwards by Nancy Feiner and colleagues where they looked at Podarcis muralis adapted to cold in parallel different populations. And they looked at gene expression in the developing embryo, and they also found a similar pattern of gene expression as this kind of differential cold adaptation happened in those populations. And then additional evidence, I wanted to also know whether the differences in phenotype that we observed, so the different bone density, for example, has anything to do with any of the genes we identified. And there was one that popped out, which was CALP-R. CALP-R is known in humans, the calcitonin receptor, to grow bones during development. And if the CALP-R receptor is not working, we get a genetic disorder called campomelic dysplasia, it's either fatal, where the bones don't fuse in the body. Secondly, CALP-R is known to be involved in osteoporosis, so the expression of CALP-R changes bone density can change quite rapidly during aging as well. So that was another evidence that possibly, we've got the right thing here. Okay, yes, but one hypothesis and one proof of the hypothesis is nice, but why would you stop at one if you can have more? I was luckily involved in this nice study, which was led by Johannes Müller and Miguel Vences, and also Juan Garcia Porta and Ike Zari were really involved into getting that together when we looked at a new phylogeny of the lacertid lizards. So there was a tree that was made with the legacy markers of a lot of species, and then additionally we did RNA sequencing to construct a phylogeny from about 7,000 genes of 24 species. It was actually 24 plus 2 divergent lineages. And in conjunction with that genetic sampling, the collaborators, they measured also the preferred temperature in these little experiments here, and also evaporative water loss rates. And then I took that data set later, after we published this paper, to test for genomic signatures of selection. First of all, an interesting pattern that popped up here is that we found that the molecular substitution rate, so the way or the rate genes change over time, actually was really correlated with the climate that those species were inhabiting. So we have 7,000 genes, and if you look at all of the 7,000 genes, they kind of progressively evolve slower the more the species kind of evolve towards the north and the south poles. So the lacertid lizards originate somewhere in a warm climate, and then they did like a poleward expansion during their evolution, and as that happened, the molecular substitution rates kind of dropped down. So because it was a northward expansion, you can see here the temperature graph, you can kind of read it like an arrow going down this way. All right, so interestingly, climate influences rates of evolution of genes, which made me very excited because I never seen anything like that. Okay, so then the test of selection, instead of 7,000, that was just now 6,000 genes. I had to take out a few in terms of quality control. And we were looking at, okay, where can you find significant kind of evidence of selection? And there was 200 genes that came out. I'm not making this number up, it was literally like a round number. And many of those genes had, again, I was looking at the functions, many of those genes had functions that were related to reactive oxygen species, mitigation processes, and also they were related to mitochondria. And mitochondrial activity regulation is one of the classic kind of ways species adapt to cold climates. So that's again kind of an evidence that adaptation to cold climates is important in species that have expanded towards colder climates in their evolution. And we found a few evidences for heat-specific adaptation. Possibly there could be some alleles in HSP90 in Helena-Lacerta greca, but that was just that one species. However, there was still kind of some evidence for adaptation to warm areas in the species that are inhabiting those warm areas. Okay, so again, let's look at our candidate functions. And the 200 genes of the lacerted lizards are shown on the right. And yes, we did cover some candidate functions, not all of them, but also we didn't cover any other functions. So that was interesting. There was literally just a few genes that had other functions that could have been used for other purposes, I guess. All right, let's look at some genes in detail, just two of them, because it can be a bit much. So we're looking at the association of the rate of evolution of the gene with a specific type of habitat. So that's an analysis that you can do now. And basically this gene on the left here, this EIF2AK4, is a transcription factor, and that was showing significant evidence for accelerating the relative evolutionary rate in species that have adapted to hot climates. So species are all shown there in orange. And then we have another gene, MRPL48. That one shows a significant deceleration of its evolutionary rate in conjunction with the evolution of species that are living in cool climates. And that's one of those mitochondrial proteins up here, so that makes total sense. And then I was looking at this EIF2AK14, and it turns out that it is already known to be associated with heat adaptation in different species because it helps regulate blood vessel size and circulation, which helps sweating or exchanging heat with the environment. And specifically that was studied in Chinese cattle, so it already helps to adapt Chinese cattle to heat stress, and possibly also inserted lizards to live in hot environments. Okay, so this kept popping up more and more. So I'm looking at this and in the literature as well, and there are lots of instances where you find one gene to be discussed to be really important in the species to adapt to a certain climate, and then there's a paper about a completely different species pointing out the same gene. And one of the most famous ones, I think, in this instance is ADAM17. It's a metalloprotease, which helps to kind of hemoglobin to deal with low oxygen levels. And that gene has been found to be really important in Tibetan humans, so in high elevations, but also in the yak, which is also living in Tibet in high elevations, but also in the ducks that migrate to these areas, and also in the dogs of people that live there. So it's like in 4 different species, the same gene modified to kind of deal with this kind of environment. And the question is like, is this just a coincidence, or is maybe the number of genes that can help organisms adapt to climate actually smaller than we think? And the second example is from Enola cybotis. So if I pull out some of the genes that we were thinking is maybe important in climate adaptation to cold climates, we found the same genes in a study that was comparing the mammoth to its kind of alive relatives. And it uses the exact same, it had like modifications in the exact same number of genes. And it was speculated that that helped it to adapt to the ice-age environment. So that was a question, of course, that can't leave unanswered. So we went again and we did a bit of literature research. And luckily, like in the meantime, a couple of years after all of this work, there were a lot of studies published on adaptation to environments. And that could have been comparing species or populations along environmental gradients, or it could be two sister species that inhabit different habitat types, or maybe, like a fish that lives in a completely normal spring and one that lives in a sulfur spring. And we just collected that information for many different abiotic parameters. And I always get kind of hung up on the fact that I sometimes confuse abiotic and climate. So we analyzed abiotic, but I'm interpreting here for this talk mostly climate. Okay, so that was 25 studies of genomic adaptations to climate, and that included the certity, like the genes that we just found, plus other ectotherms, plus also some endotherms, because the data was just there. And I also wanted to check, is there a difference between physiological strategies? So do you use different genes if you're a mammal or if you are an ectotherm? And then also we looked at 75 studies on the transcriptomics of the environmental stress response. So if you have just one species or one population, you expose it to heat or cold, any of those things. Yeah, so that was basically a companion data set because we wanted to know what's the difference between adaptation and stress response as well. All right, in all of this literature searches, we ended up with 1,100 genes. Again, a very nice straight number, easy to remember. Which was actually way less than we were expecting, because if you can imagine, like an average vertebrate genome has maybe 20,000 genes in it. And if you look at 25 studies of different species, you would expect quite a lot of genes to be like doing this function, but it was just 1,100. And on top of that, about half of those, like 43.6%, were performing this kind of function in more than one species. So almost half of the genes were reused more than once, and it can be almost like 4 or 5 times, so 4 or 5 different species using the same genes to adapt to different environmental conditions. And then, interestingly, what we can look at is this little graph over here. I'm apologizing that it's a bit pale. I hope you can see it. The good thing is that for lucidity, we also have this preferred temperature that we measured experimentally. And you can see that the genes that adapt to climate or environmental change in more than one species in lucidity are actually much stronger associated with preferred temperature. So this is again this variable that looks at the association between evolutionary rate and the habitat the species live in. Yeah, so if the gene is used more than once in vertebrates in lucidity, it's stronger associated with preferred temperature. And then on top of that, 18% of those genes were also in the environmental stress response, which I think is kind of important to say the opposite thing, which is that not all of the genes we found were involved in the stress response. So reviewers rightfully said, well, 43.6% is like the number 42. So what does it actually tell us? What's kind of the random number of genes that you can expect to find more than once? If you look at enough studies, so maybe if you're looking at all the studies in the world, you will always find like the same genes because there aren't that many. So we did like a simulation which is called the birthday problem, which is basically a modification of the question, what's the chance that any 2 people in this room have birthday on the same day? And so what we asked is if you take a genome of 20,000 genes, what are the chances of finding exactly 43.6% of those twice if you look at so and so many studies, so if you ask so and so many times, right? So we looked at our 1,843 studies and the birthday problem simulation. We found that we would have had to look at 4,800 studies to find this number just by chance in a genome of 20,000 genes, or even narrow down a little bit more in 9,840 genes that have those same general functions that I just showed. So it's not just about functions being general, it's apparently there's actually something there. Okay, again, our little table, so which candidate functions are covered by those genes, and it was all of them, but there was another one which we hadn't looked at, the cytoskeleton. I have no idea what the cytoskeleton does in the evolution of vertebrates, genomes, but it's apparently important, but it's definitely not canon. All right, good. So, summarizing that, from looking at lots of studies and doing our own studies and things like that, we found that over only a few genes and functions seem to adapt to different abiotic environmental parameters. Most of that data was climate and some other abiotic parameters as well. And we have the occurrence of frequent reuse in very distantly related organisms as well. The physiology or stress response is important, but there are also other general functions where we can't really tell yet why. So why is membrane transport important? Maybe we'll find out experimentally at some point. And I think in the lizard examples that I've analyzed so far, most of this adaptation is actually to cold climate and not to warm climate, which makes sense if you look at the circumstances that those different ecotypes sort of evolved in, either the cooling Cenozoic or going up a mountain in the Caribbean. Okay, so now it's going to be a little bit more theoretical. We're going to talk about networks. So I think if you're like, ecology adjacent, you probably have seen networks pop up quite a bit lately in terms of people looking at predator-prey networks or ecological networks or even like community networks. It's kind of a new way of thinking about data. And the same thing is happening in genetics as well. So originally, networks have been invented by, I guess, cancer biologists that are looking at, co-expression of different genes in a certain condition. Or, if one gene, does something, how does it affect others? And I was very fortunate to pop into some places where people were actually doing this. And I was thinking, oh, how can we actually use this technology or use this kind of new approach to understand a little bit more about questions that are relevant to ecology, biology in general. So if you're really not really that much into genetics, and maybe yesterday was a bit too long, I'm going to show a lot of figures that look a bit like the exploding Death Star. So, like I said, if you kind of want to check out a little bit, you can just look at the figures, enjoy yourself, and think of Star Wars. But yeah, for everybody else, let's do some theory. Okay, so we have to define two things. So we have to define constraint. Most of you have heard constraint before. I've mentioned it a few times before, but I think I'm going to use the definition of Stephen Jay Gould when he was pondering the Ice Age deer skeleton. Constraint is defined here as any factor that limits the potential of natural selection. So if you think of evolution as a process that always flows and flows and flows, constraint is anything that kind of holds this back a little bit. Okay? What it doesn't imply is it goes a certain way, like it's kind of going in a certain direction. That's predefined. That's not what it is. It's literally just anything that holds selection back in some way. And the second thing that we need to define here is because ecologists have different understanding of this, is pleiotropy, but on a genetic level. So typically pleiotropy is you have one gene and then you have an effect if you change it in multiple traits. So multiple traits are different, and that's what we call pleiotropy. What I define as pleiotropy is you have one gene product, and it affects other gene products in pathways, and then in the end you have multiple traits, but that's not really what I'm looking at. And also I don't know, so if I know a gene interacts with another one, that means in no way that I have any idea what comes out in the phenotype afterwards. So that's just, a little caveat here. I really, I can't tell all the time. Okay, so again, a functional network and constraint in that regard. It's just the fact that we're looking at genes now as working together with other genes. So your typical genetic study back in the days consisted of looking at one protein or one allozyme, and then we look at one SNP, and then we look at several SNPs, and so on. But now we also want to look at how actually the products of those genes, like the proteins or the small RNAs and things like that, how they work together to perform the function of being the organism in different situations. Yeah, and the interesting thing is that the network that comes out of this has this thing called emerging properties. It means that its functions is more than just the sum of its genes. So there's something new here that just occurs by the fact of this being a network. And the question that I was asking, in that respect was, how do emerging properties such as pleiotropy, gene working with other ones, influence climate adaptation and maybe constrain it also? So yeah, a couple more slides like this, and then I'm going to tell you how to actually use it. Good. So again, gene products work together to perform functions, and there's a few studies lately popping up. So 2023, if you look in them up, they literally published like a couple of weeks ago, where other folks have sat down and said, well, but things are connected, how does this influence climate adaptation? There was one paper about looking at adaptation to temperature, and these authors said that, well, genes that adapt to temperature probably also are related to other stressors, such as metal pollution, and how, if you adapt to temperature, can you still respond to this other stressor? How does this work? There might be a constraint here, so they mentioned genetic constraints. And the second study was a simulation study where they looked at thermal performance curves, which means that performance is best at one certain temperature and then kind of falls off. Many of us have seen those. if you kind of add constraints to that, so they can't just vary whatever they like if the temperature changes, that can actually help the population to maintain itself and lower extinction risk. That's again a bit abstract, but that was a simulation study, so things like that are always really cool to look at as well. Okay, so again, for those of you who just woke up, we have a network, we have genes, that is like little bubbles, and then the sticks between the bubbles are arrows, those are the functional connections between them, yeah, ball and stick. So, how does this actually look like? So, we have in the middle here something really cool that also just popped up last year. It's the first 3D model of any cell. All of the products of the cell, all of the chromatin are the little loops here, and all of the proteins are the little balls here, and that is a mycoplasma. So mycoplasma is like the smallest cell that can exist, the lowest number of genes, except for those engineered ones. And you can see like how full this is. So this doesn't look like a typical biotextbook now, does it? Where you have like a kidney-shaped thing with like 3 things in the middle, and the endoplasmic reticulum and things floating about. This is how this actually looks like. So what this looks like to me is almost like a mechanical computer. So you have little things that kind of ratchet around other things, and all of this moves as well. Have you seen The Secret Life of the Cell? It's a really cool video where you can see like how all of the things can move. And then they solve problems as they do that. So it's like a little analog computer. And then you can depict that in form of a network. So you take all of the gene products, you draw down all of the functions they perform with others, and then you plot it as a exploding Death Star. So this is how this looks like. In this case, this is a protein-protein interaction network. There are many others, so you can have like microRNA networks and transcription factor networks. But I'm not just going to go there. It's just like, if I do this with geneticists, they're always like, but what about the RNA? Not now. Okay, so I set out in 2020 to test whether maybe this has something to do with evolution as well. So just the fact that some genes are connected with others more than other genes are connected with others. So maybe the question was the fact that genes can accumulate variation. Is this possibly determined by the structure of this functional genetic network? If it is, that means that just having this element of constraint, and then remember slow down evolution, means that the number of and position of genes in this network that can actually evolve over time might be limited a bit more than what we think it is. And that would be great because, there's always this kind of problem that, lots of different combinations of everything is possible, and how can, evolution produce similar genes popping up several times if there are so many possible combinations? Maybe there aren't that many possible combinations. So what I went out to test is I went to split this network into 3 parts because it was just easier to think about. So I have the hub nodes, the intermediate nodes, and the peripheral nodes. We're going from inside to outside. And the idea was that hub nodes are the central nodes. They have highest constraint because they're the most important for the organism to function, but they have less connections than the second category. So these ones have intermediate constraint. Constraint should taper off towards the outside. However, pleiotropy, which is the connections to other genes, in my definition, is highest in the middle. Yeah, and then on the outside, you have the peripheral nodes of genes. Those are the lowest constrained ones that have the least connections. If you plot this like that and think about it, you come up with the idea that adaptations should be most frequent in intermediate nodes. Right, if you want to read more about why, there's an entire theory paper you can go through. Okay, last theory slide. Basically, it's really easy to calculate that because there are already like softwares and programs that can calculate you the statistical value of each gene inside that network. So you can find out which ones of those are hub nodes, intermediate nodes, and peripheral nodes, download that as a table, and then go crazy with statistics. So that was really good to be able to do that. And I tested that hypothesis first in yeast. That's why there's a yeast up there, not a lizard, so sorry. But we'll get there. So I was looking at different strains of yeast that are using genes to adapt to differences in their environment. So that could be like food or something. And I found out that, as expected, those genes, the ones that are reused by more than one species of yeast, are actually in those intermediate nodes as well. So they look like they're in the center, but they're actually not. They're on this kind of intermediate shell around it. Well, let's look at mammals real quick, and real quick because a couple of months ago they published this paper from the Zoonomia Consortium where they looked at 265 mammal genomes and calculated for every single gene in the mammal genomes under which type of evolutionary constraint it is. So I just took the data, put it in a network, and the same thing comes out. So constraint is highest in the center of the network and then tapers off towards the outside. That network is human. You can see the same thing here with the different categories of nodes. So constraint highest in the inside and then goes down. And I was looking at, well, where does hibernation occur? So if you're a mammal and you want to hibernate, which genes do you use? And luckily, they also calculated the association of evolutionary rate with that hibernation phenotype. And I plotted it on the network, and it was also in the intermediate nodes. This is 3D, so some of them you can't actually see that well, and I just wanted to avoid making it look like it's the center. It's actually on that same shell, kind of towards the back. And there are some key nodes as well. Yeah. Right, back to the cells. So, good thing is we're getting more and more genomes, and I'm really excited about the fact that we're getting genomes from non-model amphibians and reptiles now, where we can look at all of those interesting questions on more than like your typical standard species. So in the Podarcis viralis network, which I plotted here, the same thing happens. Constraints highest in the center of the network, and then it tapers off towards the periphery. And why we care about this is because of our climate adaptation. So now we're bringing back that information that we had before we looked at exploding death stars about genes that adapt to different climates. And you can see here in like pale blues, maybe not the best colors, we can work on that. You can see the background nodes, the ones that we don't know if they're involved in climate adaptation or they just don't. I always use that as like a comparison one, because I guess we don't know. And then we have in cyan blue the genes that we know adapt to environmental change in vertebrates, so that's not just Lucertis. And then we have in yellow the genes doing that in more than one species. And of course the question is, well, you know, are they organized in that network in a certain way as well? And if so, why do we care? What can you tell us? Yes, they are. So again, no genes of environmental adaptation in hub nodes. There are some in peripheral nodes, but most of them are in the intermediate nodes again. And it's just, sorry, this is just a graph that I pulled really quick because I'm not really done with that yet. That's just a histogram instead of a box plot. And it shows you that basically it doesn't matter if they're in the intermediate or in the peripheral nodes, the genes that are used for environmental adaptation always have a higher constraint than the background nodes. So that means that those genes are not essential, so they're not sitting in the hub nodes, but they're still important, they're still constrained, and they're constrained because they have a lower evolutionary rate, and they are also connected to many other genes. That means that they're likely going to have a bigger effect in the phenotype as well. Okay, so let's look at our Lucertis. So these are just the Lucertis genes, and again, we're looking at this, this metric that tells us about how strong the gene is kind of related between its evolutionary rate and the warmth of its habitat. And we see that genes that are strongly associated with warm habitats are also located in those I and P nodes. Yeah, so I and P nodes evolve fast in association with warm receptive habitats. So we have a, sorry, this was sliding around, I don't know why. So there's like this constraint, which apparently influences how well certain genes can help the organism to adapt to different environments. Okay, so let's summarize this, because time is running. This is just me speculating, so there's no paper and prep or anything underneath that. I'm just trying to explain it to you. So what I'm thinking is, if you're looking at any genomic network, in this case, Podarcis viralis and the environmental adaptation genes, you have in the middle always this protected zone. So those are the genes that are important to develop the organisms, like Hox genes, things like that. And mutations in those are rare, just simply because any zygote that would carry a mutation, they would be immediately selected out. So they wouldn't even be born or anything. It just wouldn't happen. Second, you have like this outer shell where mutations are common. However, they don't have a big effect on the phenotype through selection because they just have very few connections to other genes. If you have, for example, like an opsin, it's just expressed in that one, adult life stage in the eye, doing one function. It doesn't really interact with that many other ones. Right? Maybe not like the stress response genes, which are the physiology of the organism. And then you have the intermediate shell, which is basically the place where things happen. So you have a mutation can happen because it's not super constrained. At the same time, pleiotropy means there's going to be a big effect if it happens. And you can then actually make a big difference in the phenotype. Okay, so I think that's where this happens, and that's why we have all of these genes concentrated over there. Okay, take-home message, functional network constraints can explain evolutionary outcomes. So what happens in the genes and in evolution can have something to do with the network. Environmental adaptation happens mostly in the intermediate part of the network. Those genes are less tolerant to mutations than others, but they're more connected and thus have a phenotypic effect. And we could use this to estimate which genes could contribute to future climate adaptation. All right, so things happen very quickly. How do we keep pace? This is the figure that was published in this nice new review. It's pretty much the same as my badly drawn rainfall picture. There's a fine line, which is the diagonal between the population being stable and the population crashing. That is an outcome of the trait values, what happens with the trait, and the time axis, so over different generations. In the basal trait values, so the traits as they are right now, as climate changes, and the generations go on, the stable population will start declining very, very quickly. And there are different ways of keeping pace with that. One is employing plasticity, like behavioral or genetic plasticity, which can shield variation from selection, but on its own, over time, will be insufficient to keep pace. And there are different mechanisms that can contribute to keeping pace, and those are all microevolution, which are either using the standing variation that the population has to select for a better phenotype kind of distribution, or it could also be changing the evolutionary limits. So there could be a role for novel mutations here. That, if you think back about the lacertic graph, in warm habitats, evolutionary rate of gene goes up. So there could be something here in terms of mutation rates. That's what my new project is going to look at. So I'm going to look at, with my team, 3 larval ectoderms. We're going to look at the tadpoles of this Heteroxylus Madagascar reed frog, a marine ragworm, so completely different organisms, because I like to jump around between them anyway, and zebrafish larvae. And we're going to look at how different kind of stressors, such as heat stress, influence the ability of organisms to repair their DNA against breakages, and what that means on a sort of micro-evolutionary level. Yeah, so overall, I think we're going to hear a few very, very interesting talks in this symposium. What should we do to keep pace with climate change, which, like I said, is happening right now? The birds are falling out of the sky. We need to do more research on, first of all, mortality events. We need to figure out what's the effectiveness of plasticity and what are the trade-offs in terms of genetic and behavioral plasticity as well. What are the effects of migration? So, how can organisms shift their distribution area? What are the obstacles? What are the physiological limits? And it's surprising how little we know about many species. There's a few that we know a whole lot of, and there are many species that we know nothing about. And then we can go to my student Ed's talk in the same symposium here at 3:20. How does the stress response even behave in different species, in different environments, and how will it adapt? And then lastly, I'm hoping that today kind of made you a little bit interested in functional and comparative genomics. What can we do sort of from a bird's eye perspective to help understand this process a little bit better? All right, so in conclusion, the pace and direction of climate change are unprecedented. It's going in a different direction than it's gone the last million years before that. The genes that respond to this are few and have defined functions, and combined with network positions, we may be able to predict where adaptation can take place. However, as a practitioner-oriented person, you might be thinking, what on earth could this be good for? Well, let's have a look at our friends, the coral biologists. So the coral biologists are freaking out right now because their corals are bleaching rapidly and dying. So there are a lot of papers that I handle, for example, or that I've seen published lately, where people look at how can we make those coral symbionts withstand heat a little better. And something that they do is they screw around on the HR protein 70 with gene editing, or they just do mutagenesis. This paper came out a couple of days ago, where they just blast UV on top of the coral symbionts and just see over time, maybe we'll find one that's a bit better, suitable to withstand this heat stress. What we can use, like what I've just talked about for, is first of all, looking for a standing variation to select from that actually matters, yeah? And also maybe better targeting these mutagenesis studies. I found a few genes that are published that no one's looked at, but they're involved in literally every single stress response experiment. And you find it in the transcriptome, but it's never being discussed because people haven't had a look at it before, right? So you look at these comparative data sets, and that can help you focus on things that we maybe haven't focused on before. And second of all is I hear a lot about heterozygosity. So I heard a few talks about how this is important, that it's high because it can help adapt. But maybe we have to look at heterozygosity and where it sits in the network to see which one of that heterozygosity that we're looking at actually matters, which is the one that can be used to adapt to these new environments. Okay. So here are two QR codes that I would like you to take a picture of and go to. This is me advocating for the new Amphibian Genomics Consortium. There wasn't any amphibians in this talk, but a few folks got together from the University of Melbourne Veterinary School to just take a stock of which amphibian genomes do we have, which ones do we need, and how can we do some capacity building to improve this. So, I'll just put this in there on behalf of the organizers. If you are working on transcriptomics of stress response, or maybe even some selection studies on genes, you can submit that to our very kind of provisionary database. It's on our website. You can also, she has a Twitter handle, Cassandra. It's just a Google form, but everybody can go and download those genes and what they do and the functions in the most current version that we have. And like I said, I'm going to go in the future to kind of give this a better address and then pad it up with newer studies and keep this like a resource for the community going forward. All right, thanks for many collaborators, more than I could put on the slide. Thanks to the funders and thanks to you all for listening.
Fantastic talk. Thank you so much. Any questions from the audience? Trent.
That was a brilliant talk. Thank you very much for that. That was really cool.
Like
and I apologize in advance if my questions stupid because of my lack of understanding. But one of the things that struck me about that when you're doing the network analysis thing at the end is there's a bit of a numbers game there. So lots of links means lots of genes means lots of importance. But if I were to turn it on your head and actually use your example of opsin, no opsin is pretty bad, right? It's like a few links. Is there any way to interrogate your networks to say that there might actually be disproportionate effects based on certain genes that are independent of all the number of network links?
Yes, so I had written a grant about this, sadly it got rejected so many years ago, and the idea was looking at, sort of, like you said, more those central things that influence lots of traits, but also the ones kind of towards the outskirts of the network. And I was hypothesizing is that those kind of modifications you find mostly in single species or populations. Like that's kind of your key innovation. I mean, of course, losing something is always bad most of the time. But evolving something new, if that happens only like in selected places and it's always a different gene, I was expecting that it kind of sits more towards the outskirts of the network. But like I said, I haven't been able to test that yet. Might go back to one day.
Thank you. Any other questions?
So it's the ideal talk for Friday morning, I always think. But going from the micro scale, which is looking to the macro scale, I kind of thinking that we conserve ecosystems, we conserve species, but we should be designing nature reserves and nature networks, macro networks, based on conserving or at least safeguarding some of this genetic diversity.
Yes, definitely. That's why I've been wanting to write a paper about this, because there was a paper about genetic diversity not so long ago, and like the importance of that for conservation planning. And I'm just sitting there and I'm thinking, yes, but you need a bit more information. But then how do you get that information provided to the people where it matters? And the good thing about this network stuff is that it seems to be really general. It doesn't matter if I apply it to a yeast or to a lizard or to a human. So if it's general, then maybe you could just say, well, this is a general list of things you need to look out for. Do a screen for genetic diversity, but don't just look at the heterozygosity overall to look at the health of your population, but also look if there's any variation in the gene, so. It's just kind of, breaking it down a bit more to the individual gene level. Like, I know it's really difficult to say, I'm lab computer biologist, conservation person, here's something useful for you to know. But yeah, I'm really interested in seeing like how this can be applied. And I think that's also my job, I would say, to do in the next couple of years. And I definitely, I'm thinking about it, yes.
Any other questions? Yes.
So a lot of the very interesting talk, thanks very much. A lot of the analyses that you presented, it seems like you're kind of looking at mutations or changes through an adaptive lens, but you also mentioned earlier in the talk that there are other things that can explain these changes, like neutral processes like drift. Are there any methods that are applied in these types of analyses that can differentiate adaptation from non-adaptation, non-adaptive change?
Yes, so that's why I went with the functional approach. Because yes, you get always like drift results, and there are always, divergent markers doing this and that and everything, but they might be like completely different somewhere in the genome. They might not be in the functions that actually matter. So I had a lot of discussions with people that were like, but you need to look at amino acid level, not just look at the gene level. You need to look at the base pair level just to suss out like which of that variation is like actually doing something. And yes, I totally agree, but I'm just one person, so I was happy to kind of, I'm happy to just do the balls and sticks and genes for now, but yes, so that's all, part of the thing. So if I do an experiment, we have one multiple generation sort of adaptation experiment in the lab, 2 different oceans running right now since 3 years or so. And the idea there was like, use really big populations with several replicates. And if something happens in more than 2 replicates, possibly it's not because of genetic drift. So I see the problem. But,
do people do things like with your focal genes that you're interested in that could be related to climate, comparing them to other genes that may not be, like looking for differences in substitution rate, things like that?
Yes, exactly. Yeah, I've done that a few times as well. And that's also that's on the agenda, F back there, the last row, is sequencing a whole lot of those putative climate adaptation genes across different businesses as well. But yeah, very good point. I mean, yes, of course.
Any last questions? No. Thank you so much, Ken. Before you go, I would just like to remind you, if you're presenting today, please make sure to email us your talks, and you can go and have a coffee now. Thank you for coming. Oh, yes.